The overall goal of this method is to label and visualize live chromosomes in mitotic cells using Histone2B-GFP BacMam 2.0 label and a spinning disk confocal microscopy system.
This method can be used to visualize mitotic chromosomes in cultured cells to study the effects of genetic modifications or drug treatments on chromosome segregation. The main advantage of this technique is that it provides an easy and convenient method to label and visualize live chromosomes in mitotic cells. Demonstrating the live imaging procedure will be Dr. Kazuyo Takeda, who maintains the Microscopy and Imaging Core facility in the Division of Viral Products at the FDA Center for Biologics Evaluation and Research.
The mouse embryonic fibroblasts, or MEFs, used in this experiment are grown in a 2-well chambered cover glass at 37 degrees Celsius and 5% CO2 overnight, as described in the text protocol. On the following day, synchronize the MEFs in G0-G1 phase by incubating in DMEM F12 growth medium containing 0.1% FBS. On the day of labeling, prepare a 1.5 mg per milliliter stock solution of nocodazole in DMSO.
A critical step is to calculate the right amount of CLHV particles per cells to achieve enough labeling for chromosome visualization without toxicity to cells. We had previously determined by titration experiments that 30 ppc works best.